Concentration of the
I don't know if I
fun
can
ones,
handle
like,
that
so the boiling is longer because there's a higher concentration.
Yeah. I think it's like honestly a lot of people for tonight. And I don't think it's gonna.
I forgot all right.
That was the mistakes that I kept. It's I thought and I told you I was, and like, what I actually called it.
I'm training though. Oh, you are? So many. Oh, so then you won't even be, you don't even have to stay for both of them. You said I can get out. Yeah. Which they usually close it at like 8:30 anyways, and be out. You just brought.
Mine looks a lot.
So all you have to do is close the deli.
Hey, at least you
and
did
then
it.
you can say.
Mine is white.
Mine's, I didn't think I did the right amount of sides.
There's doors hanging on.
That's what
It's
it
really like, if you have people on deli, it's because you only have two sides of the Hey. I don't know, he schedules.
I don't even think he looks at. I just don't have the energy.
I just don't wanna touch cookies.
Yeah, my-
It's
It's a true.
It's fine. It's like not focused on concentration at all.
If you ever really need one, because
No, but
a game
I
or
feel like
something,
right now,
I want
I
one, like I don't, it just feels wrong, and I still could, it's stupid, like, I don't know, it's like
I think it's a lot better to be
I just don't even cook time.
I always feel like I'm like, last time we was 2 to 3 times.
Just think it's good to put some nice food.
Okay, so I was like,
I just don't know how
I think I probably
So he doesn't say yes to that money, I was like,
Does,
what's the boss is not, cause I think I can only work like 2 days.
Wait, why did you get mine?
I don't like my bush.
Oh my god.
So like the fact.
I mean, that's
I didn't
fine,
say
but I'm like,
a single thing right. She couldn't give me any.
So I got that one right.
he's such a goofy guy.
That's the one that I got right.
Just like
So my brother is from like Blake. Mine's really. Like I understand. And
She didn't even like my little
and
shit.
like literally.
So much smarter, but now
Literally,
it's
no,
That's like the only thing I do, but that's just like pure
I got
memorization.
those.
Like the same, I
It's actually crazy. I was
Actually, really tragic, and I
Saturday, like sent a message like, hey, like,
So
that's gonna be there, and then he yelled at us.
no, it is
Just like
the way like
I have called and died. Everybody.
Yeah.
And then I gave
He's like, you should look at these fans.
Yeah, this is how you realize that. I really don't. I can't crash into for a your kind donation.
I could just figure out for you.
It's okay.
Did you know?
It's scheduled for.
Let me just double check some Mary Browder, but she has breakfast tomorrow morning at 5:30. She said we could eat. I told you that too.
Thursday night.
Still doing it. Which.
Yeah, I mean,
But I don't think it'll help.
So I was like, anxious.
I think I'm gonna have purple.
Yellow purple hair, purple
I was joking about we need to get one.
I was like, well, I guess you have to get
I've never heard.
Alright guys, let's get started. So quick reminder for next week, on Monday, you guys turn in the writer for your papers. If your group already decided what paper you're gonna work on, email me so I know which one that your group's gonna take care of. That way I can start reading them myself over the weekend. I believe I did upload the slides telling you guys how to break down the papers, right, where you guys can read out the information. Normally the abstract is the biggest tool that we use. Then after it kind of that'll guide you what's the biggest project, what's the key findings, you go to the results and really break down what they're doing. If you want to go over the techniques, you have any questions about like, what do the results mean or how do they pull the data from them, feel free to come stop by office hours. If you can't make it during my time, just send me an email, we can schedule something, we can have a quick chat. If you want to bring your whole group, that sounds good, we can have a quick chat every time. I'm looking forward to reading those, so hopefully, you guys got time to work on them. For the midterm, I barely started grading them. I looked at the keyword chart and then I got tired. So a lot.
How did that look?
Hit and miss.
Okay.
Hit and miss. So there, some of them look good, some of them they were like, I know it was kind of like, I almost picked you guys, it's a little too close, right? Like the enhancer promoter, I was like, switch them back, and somebody wrote them properly, deleted them, and then wrote them backward. I was like, no. Yeah, there was a few of them. Yeah, I'm gonna go into the to the text part, the bigger questions. I haven't done that yet. The only one I did look at was chymotrypsin, because that was one of the ones where I was like, I didn't tell you guys to study it that much, but I did talk a lot about it in class. So I was like, all right, let me see how you guys did. So there, what were the options that you guys could talk about? You guys remember?
Hydrophobic binding pockets?
And what was the other thing?
The active side.
The active side, right? So if you brought up both, A++, right? If you give me at least one A, right? So that's kind of what I wanted. So I saw that a lot of those, they were bringing up the topics of the binding pockets. Some of them didn't say binding pocket, but they were like, no, it has hydrophobic region. I was like, cool, that's what we want. So that made me happy. So I'm like, if that did good, I'm thinking the rest of it's gonna be good. It's Valentine's Day, we got time to grade. I have a presentation on next week, so I have to finish my slides and look pretty for that. So with that being said, on Monday, it's my interview day, so we won't have class on Monday. The thing is, we still want to cover, let me see. The 19th, right? So for DNA replication, one of the things that I can do is either I'm gonna shift everything down, and then during this Friday, what I can do is I can make it almost like a test prep day. So basically, I'll make maybe 4 or 6 questions. Then you guys come into class, break off into groups, and as a group, try to answer the questions. And then my goal is to make questions in the same style that I'm gonna present to you for the midterm for that section, yeah? Because the cell cycle, today, like we're gonna go over it, we're just gonna go over what is cell division, right? How the cells split up. But tomorrow, Friday's lecture, I'm gonna go super heavy in what are the mechanisms of how we're gonna regulate this, right? How do we have really tight control? So again, you guys are gonna have to get into that habit of like understanding the whole pathway and then the little rules that kind of help guide it to make it function properly. Yeah, so I think we can do that on that Monday as a group project. The other one would be, I think that's like kind of one of my best options. And then we'll basically just review. Well then, I might cancel this. You know, like normally this is the day that we kind of take time together as a group to kind of find your paper. I almost want you guys to do that. Like I'll break it into groups again, and then if you guys do that on your own, like, just take some time out of the lab to go find the paper together. You'll bring it up to me and I'll confirm it and give you some feedback. But that one we can kind of keep this review day, right? If you guys still want to do the primary literature in the class, like we did last time, then what I'll have to do is I might do like a later review session or pick it at a different time. Yeah. So I'll send out one of those job forms again. You guys tell me what you guys want to do, and then we'll figure it out. So we'll talk about that. For Monday, come on in, do the group work stuff. I'll have it count a lot more than the other quizzes, so it'll be more than 2 points. And then, it's gonna be more quizzes that we can average out at the end, yeah. I think that's my big stuff, Monday. Yeah, so Monday, don't forget the papers, I'm not gonna be here, the group work, and, let's get into it, guys. All right, so cell division, this one. We're gonna start off like we did in the other classes. Oh, sorry, I've been changing my slides. I'm trying to include the stuff that I put in for your review stuff. So like text summaries are here. I put the vocab all the way in the back, and I'm putting yellow slides to be tables that kind of summarize our info. Yeah.
Going back to Monday, do we still have class? Or no?
So I'm gonna I'll send the email out. You guys can do that group sheet as a group. If you
Okay,
want to do it as a group, if you want to do it on your own, I guess I'll just do it on your own. No,
okay,
and
okay,
then
so we'll
you don't
still
have to come
have,
to class and do it whenever you want.
okay.
Um, you know what, I'll put that in the survey, if you want to come to class, then whoever says yes, I'll group you guys, whoever doesn't wanna come to class, then you're kind of like working on your own, or, find your group to work outside. Yeah. Is that fair? Alright, cool. Alright, so let's get into the cell cycle, right? So here, what's the big thing? We're trying to get cells to divide. So the whole purpose of cell division is you're trying to copy something, right? So you have the original, and you're trying to make an exact duplicate. So if we go back thinking about, what is it called? The most basic thing that we need is we need to copy all of our information. Right? So that means that our DNA, however much we start out with, we need to have the exact same amount when we're done. The next part is kind of what we talk about, like, we have different structures in our cells, and we have different organelles, and we have different proteins, right? So nucleus is a big one, right? Our ER, our Golgi, our mitochondria, all these organelles have a function that in our integral we have in our original cell, and we need to make sure they show up in the following cell. Right? So it's like when we're dealing with this pathway, we have to make sure that all our cytoplasmic content gets through. Right? And we have to make extra copies so that each cell can again be an exact copy of the original, and it can support it. Okay? So with that being said, we're going to go through the route of I'm going to focus on mitosis. We're going to go through that mitosis pathway multiple times. And each time I'm kind of emphasize different aspects as we go through the same cycle, but just so you guys know, it's going to be the same cycle over and over. All right. So when we're talking about DNA, what I want you guys to keep track of is going to be these N values, right? So for this one, it means that, for example, one of our cells is considered 2N because we're diploids. So we have 2 copies, right? We normally get 23 chromosomes from our mom, 22 chromosomes from my dad, and that's how we make our cells. Yeah, like for us, for me, it's 22, and then of course my mom gave me an X, and my dad gave me a Y chromosome. So these would be the ones that kind of make me a little bit different, right? Everything else, like this, these would be the chromosomes. I should draw them. Never mind. I'm gonna hurt you guys, but your mom's genes, your dad's genes. At every one of these spots, they're basically, you have like an equivalent, which is something that should have the same type of genes in your mom's chromosome. You'll find almost something equivalent in your dad's chromosome, right? So these will be kind of like your homologous chromosomes. So they're kind of technically equal to each other. Like I said, the only ones that are a little different for me is that the X chromosome and Y chromosome, there's a big difference in size, right? But for X and X chromosomes, they're roughly the same size. So my cells should start off as 2N. When we go through division, so what we're going to end up doing is we have to duplicate the DNA first. So we're going to jump from this 2N phase to a 4N phase, right? So that means that I'm going to have in my cell twice as many chromosomes. At that point, I'll have 2 identical copies. This chromosome is going to get copied, this chromosome will get copied, this will get copied, and this will get copied, right? And as we then double the inside content, when the cell divides, then they go back to having $2 cells with a 2N value. I'm going to try updating my slides to making all the 2N numbers purple, because you're going to see we're going to bring them up at multiple times. And then the big difference between mitosis and meiosis, I'm not going to go that deep into meiosis here, but it's that in meiosis we start off with a regular cell with a 2N. It kind of goes through this thing of replicating itself. But then you still get cells that are 2N, And then after that, you actually divide the two end cell, split the DNA without actually copying it, and that actually ends up giving us a product of making 4 cells with just one end. And that's what gives us basically our gamete. So this would be like, could be the sperm, this would be the egg. These two then come from your mom and your dad. That's basically what you can combine to produce your 2N offspring. Yeah. But we're gonna focus today a lot on mitosis. So let's get started. All right. So for this one, the old school way to talk about mitosis, it started with just microscopy, right? So in that case, I kind of want you to be thinking about, they used to use these definitions, which was they could only see basically 2 big phases. One of them was called interphase. At this point, your DNA was free and it was just a jumbled mess inside the cell, right? There was no defined shape. So at this point, they literally just called it interphase. They thought that this was most of the life of the cell, and eventually it went to these little windows where they thought division happened, and this was considered the mitotic phase. This is where when we used to look at the microscope, you can get the really cool pictures of getting like the chromosomes condensed, right? They would see all the cool stuff of like the DNA would compact. You would see these little shapes kind of build up. They line up in a single sheet and they get pulled apart, right? So they were like super interesting phase, and they thought here it's basically like the boring phase where nothing interesting is happening. So this is the old school definition. After that, we kind of got a little bit more data on what actually was happening within this, let's say, this boring phase, and they broke it down into a little bit more information on what happened, right? So for this one, interphase kind of gets broken down into 3 different fragments. Yeah. So again, during that boring phase, the first thing, G1, what you're taking care of is your cell's actually getting bigger, right? So it's starting to replicate itself, and it'll start to grow. During this phase, there's a little bit of, if your cell is okay, the environment is okay, it might stay almost like in this, like in that environment, kind of just waiting here, and it doesn't actually have to go to the following steps. Sometimes having like a lot of resources or getting other signals can actually induce it to keep going, to try to copy it, but it can kind of sit here. When it does sit here, that's when we call this G0, right? So it'll be like an alternative concept, like if you're not gonna divide, you can wait there, or you can decide to keep going. If we decide to keep going, we're then gonna enter this S phase, or it's gonna be the synthesis phase, right? That's kind of the big, the most interesting one for me. So what we're gonna see is we're coming in as a single cell. We're still technically 2N, right? So we have 2 chromosomes. During this window, the big thing that's happening is we're gonna duplicate our DNA. So as we transition from this G1 to this S phase, by the end of this, we're gonna have twice the copy, so we should have that 4N cells, right?
Yeah. It's just 4, is that 4 chromosomes? If 2N is diploid and 2 chromosomes, 4N is.
Yeah, so that's why I'm not happy with this drawing. I'm gonna edit it. So it'll be like, let me draw with these 2, right? So it'll be like, if you're at that 2N phase, let's say this would be the chromosome we got from our mom. That's the chromosome we got from our dad, right? So this would be like that coil thing. When this is happening, your goal is you want to make an exact copy of this guy, right? So what's gonna happen is you're gonna go from, let's say that's your base, and then you're gonna make a second copy to that, like that. Then you're gonna have the same thing. This will be the homologous chromosome. So this will be the old ones. This one got copied that way, this one got copied that way, and now you're at 4N. Right? But technically, like for us, if we line them up, you know, we have 23 here from my dad, 23 from my mom. When we're done with this, we have 46 from each. So you have like, what is that, 92? Right. But and later I'll show you the names. These are going to be called, so these would be the originals. When we talk about these 2, their chromosomes should technically be super like identical to each other, right? So when these are identical, we call these sister chromatids. So this is like sisters to themselves, sisters to themselves. Between here and here, this would be the 2 different chromosomes coming from each parent. And in that case, you call them homologous chromosomes. Like they should be equivalent information. They're not going to be identical, right? This is the special information. we go through this, right, you're gonna go from this, having only one copy of the chromosome, you're gonna double it up. This is gonna be through the S phase, through the G2 phase, right? This is another one where we have additional growth. And one of the big things that you're doing in this step is that you're making proteins that are gonna help you with the division, like you're setting up the environment for it. And then finally, we get to the interesting one, right, which is the mitotic phase. Right, so, like I said before, interphase, mitotic phase. First step is they broke down how the cells actually grow and you copy its DNA, and that got broken into G1, S phase, and G2. DNA. So this one, I'm gonna bring it up. I'm gonna bring it up here and I'm gonna bring it up in the test. So one of the ways that they were able to kind of figure this out, to get the values for this, was using this technique called flow cytometry, or they call it fluorescent activated cell sorting. So the way this thing works is like you grab an organism, you can take all their cells, and you basically shred them, so each cell is by itself. You then run them through this machine, and one of the things you can do is grab a dye that binds the DNA. And when you're putting it through the machine, you can shoot a light through it, and depending on how much DNA you have, it'll give you a stronger, a higher amount of dye, which will give you a stronger signal. And you can kind of see how much DNA is present based on how bright it is. As each cell's kind of falling by itself, you can kind of detect how much signal you get from it, right? When they did this kind of data, they picked up this kind of curve like this, right? In some cases, they pick up low concentrations. This is the one where they consider this 2N, right? So this will be when we're in this phase, right? They also picked some cells here in the middle, and then you can see there's another big peak at the 4N, right? Which is where I'm getting to this one. So for this, I want you guys to feel comfortable of like the way this works, right? Going from G1, synthesis, G2, and mitosis, where would you expect to find the 2N cells? Right? G1, right? How about where will we find these intermediates? Something that's like 3N.
Synthesis.
S phase, right? So somewhere in here, if you're at the 2N and you barely started, right, you're gonna have just extra DNA, you might be like right here. If you're getting almost to the end of the S phase, you're probably gonna be closer to the 4N, right? And then, how about the 4N? Where would we find that one? G2 and mitosis, yeah. Right before you get a cytokinesis. Here you still technically what you consider one cell. The moment you hit cytokinesis, that's where you cut. Oh, yeah, yeah, that's good. All right, G1, G2N, beautiful. The video, you guys can look it up after class. All right, so now, again, we got more details about what's happening here in the, let's say, the boring phase. Now we're gonna zoom in into the fun part, right? So here we're gonna break up mitosis a little bit more. For this one, we're gonna break it up into two parts. We're gonna look up mitosis, and we're gonna break it up also into cytokinesis. So these will be these two big broad categories. For this, I need you guys to remember, these are basically the steps that we're gonna be using in the class. So prophase, prometaphase, metaphase, anaphase, and telophase. The way I remember them is, it's kind of funny to me. It's like a pee mat, like for your dog where he pees on it. So like mitosis is a pee mat, and then whenever I hear it, I never forget it, right? On midterm, I'm gonna put the picture, so if one of you messes it up, I'll be like, pee mat, kill me. But so pee mat, prophase, prometaphase, metaphase, anaphase, telophase. So we're gonna walk through these. I want you guys now to take note of what is the big effect that's happening in each one of these steps, because I'm gonna ask you questions about it later on. The other thing is Friday, when we go over how it gets regulated, you'll see that specific task that we have at each step, I'm gonna bring up a mechanism that's super critical to regulate that pathway, right? So let's get started. All right, so we'll do the first run through, which is you have your jumbled DNA, right? Hasn't been organized yet. In that prophase, you're starting to try to condense the DNA. At this point, you're already at the 4N level, right? Because we came in through here. So we're G1, so we're 4N. So when we come into this side, we're already 4N. We start condensing our DNA. As you see it, we're gonna start connecting it. We're gonna have these two sisters kind of linked up together, and we'll start lining them up. towards the middle. This is gonna be your, like your, I think it's called a metaphase plate or it's a plate, I'll remember in the picture. At that point, you're gonna split them. And the whole point is you line up these identical copies and then you put them vertically. So when you split, you make sure that you get one copy from here, one copy from here, and one copy from this one, and one copy from that one, right? After that, you're gonna kind of reassemble your nucleus, bring this back into inside, and then eventually you're gonna split it and make two cells. So that's one way we can go through the story just at the end. If we come back, now we focus on the green things, those will be our microtubules, right? This is gonna be like our big players or the big molecules that are gonna help organize this pathway. They're gonna be the mechanical force making stuff move around, right? For these guys, we're gonna be looking at these two little centrioles here. They're initially kind of trapped inside with each other, but eventually we're gonna copy them, we're gonna split them apart from each other. What they're gonna do is they're gonna build the base to make these microtubules. We're literally gonna turn off all the other microtubules in the cells, try to break them all up, and just keep these structures well formed. As they're doing that, they're gonna have two tasks. One of them is they can grab on to the center of our chromosomes, and then through that, they can mechanically move them, right? The other thing is that some of these microtubule chains that get made, they actually link up into each other. And we're gonna have motor proteins that are actually gonna help provide a force that's gonna push these two molecules away from each other. So by them working with each other, they'll push this and this to like outer edges of the cells themselves. So we're gonna keep track as we go through the process, eventually leading up to, we make these two structures, they push away from each other, but they also help organize the chromosomes. Then eventually when we're done with them, we get rid of them, you know? All right, so now we're gonna go through the actual steps slowly and not talk on each slide, but keep in mind that we're gonna be talking about these two stories throughout. All right, so let's go with the big player, right? So for this one is, the cell normally contains only one centrosome. All right. When it's sitting out here, it only needs one centrosome. It doesn't mean, it's not dividing, doesn't need a centrosome. Once we're starting to set up that we need a division to happen, that's when we decide to start making the centrosome. Right? So we'll make two of these guys. And then these guys basically consist of two proteins called centrioles. What these guys actually do is they work with microtubules. Does everybody remember a little bit about microtubules? Like what's their big thing? Big synapse, anybody? Microtubules, what do you guys remember?
They grab onto this, so they kind of pull, that was what you talked about in the first.
Yeah, so they'll grab onto things.
And then
You can
pull stuff to the-
You can use them for pulling. They're also shaped like a straw, so they're also good at pushing too. Like they have a good what's it called, compression force, right? Do you guys remember anything about their dynamics? You guys from other classes? No. So microtubules are pretty cool that they can kind of clamp onto each other, right? But it's almost like imagine like a conga line where people are dancing and they kind of like stack up. The cool thing is it has a weird dynamic where the negative end falls off, the binding and unbinding is slower. At the front end, depending on what's happening, molecules can land really fast and the chain will grow, right? But if nothing binds to it, it can also fall apart. If you build something and then you trap the end and hold it onto a protein, it makes it so stable that the molecule can't just break from the middle, like it can only break from the edges. So if you hold both edges, you can make something super stable. And that's where these guys are gonna be super powerful. Like your centrioles, they're gonna be holding on to the minus end of your microtubules. So it means that at least on one side, we're gonna make sure that this microtubule is not gonna fall apart. Then we allow it to basically grow out. I'll put a video of how they work just so you guys can appreciate a little bit more, but they're pretty cool, right? So big thing is they're gonna give us the base to build out these cables. You're minus end, you're at the centriole. All right. So here we're looking, again, at the late phase of the interface. We're kind of sitting in that G2 window. We're trying to prep this, right? There's basically, they start off near each other. There are two centrioles, and they're starting to build these cables, minus end here and positive end on this side. At this point, what we're going to start working on is when we go to the next step, which is our prophase. We're going to start, what's it called? So one of them is we're gonna play with the nucleus. We're gonna start disabling the double membrane on the nucleus itself. The other one is our chromosomes are already gonna be compacted, so they're gonna be super coiled into themselves, and the sisters are gonna be lined up with each other. Right. And then what we're gonna see is that these, basically the centrioles are gonna build these cables. They'll start pushing and they actually start linking up with each other across the sides. And they use the cables of the microtubules across each other to kind of push against each other. The longer the cable forms, these basically will link up. Like some of the ones you see kind of crossing, cutting across here. They link up into each other and actually push the two centrioles away from each other. So at the end of this, what we're gonna see is that for the next step, for the prometaphase, we're gonna have them completely apart from each other. They're gonna be using these cables connecting to each other to push against each other. They're also gonna be connected to the chromatids, right? They're gonna connect at this spot called the centriole, and that's done via this kinetochore. So here at the center of these two chromosomes, there's gonna be a little spot where they're kind of linked up into each other. This is gonna be a protein complex built up there, and a protein complex built up there. Then your kinetochore basically yields this user complex here. And that'll give us basically these extra proteins that can link up into those microtubules. So here, all right, so we're gonna finish breaking down the nucleus so it'll be completely free. We're connecting our chromosomes across the center, and then the centrosomes have completely moved to the opposite sides. So for this one, when we go to the next step, it's gonna be our metaphase. For this one, I didn't remember metaphase, I put middle, because this is when you finally end up lining everything up across the middle. So you should be able to see each sister lined up the same vertically across the middle. And it has connections to the centriole, to the kinetochore, to the microtubules. It has to be connected to both sides, right? So let's make sure that these two identical copies, you'll have one copy for one feature cell, you have another copy for the other feature cell, right? But you link up to both of them on both sides. This is actually pretty cool. In the next presentation, you'll see that when this stuff isn't working properly or it's only linked up to one side, the cell actually waits until the second linkage comes in to make sure it can grab onto both. Yes, here's a little bit more of my drawing. So for this one, the big thing, right? Everything's lined up in the middle. Then for the anaphase, this is when you're gonna start pulling them apart. You're basically gonna disable the protein that's gonna be stuck right in the middle, holding the two sister chromatids together. This allows you to then retract the microtubule from both ends, and it's gonna end up putting exactly one copy of each chromosome on each side. And we get to this phase, what we're gonna be seeing is for the telophase. This is when you, in my head, I almost think like it's already tear. At this point, you're gonna start to rebuild your nucleus, right? The chromosome's gonna, once it's inside the nucleus, there's gonna be signals that are gonna tell it that it's in a safe environment, and you'll actually start releasing it again. And then the other thing is we're now, since we already did our big separation of moving these two components apart, you're gonna see that we're gonna start to disassemble our centrioles. So we're gonna start getting rid of basically all the microtubules that we need. Here we switch in jobs. Now we're gonna be focusing on this thing called a contractile ring. So basically between the two cells, we build like a little mesh on the inside that kind of cinches on itself like that. And what it does is it pulls on the plasma membrane till it pinches off the two cells from each other. And then eventually the last step is cytokinesis. And this is when you do that little final cut, and now you have two daughter cells that have a nucleus, the DNA sitting inside, and this thing is back to having a single centriole. All right. Let me go back to this. All right, so this drawing, I kind of don't like it. I think it's a little bit different than this one, but we can kind of use it to tell us our whole story, right? So here, they're like I said, they make it sound a little bit boring when you just go from interface. Like you start with a regular cell, right? And as you go through interface, they don't tell you that much information, but now we know that's where our big stuff was happening, right? G1, our cell's getting bigger, In our S phase, probably somewhere around here, we're copying our DNA. Now we have a duplicate DNA. And in G2, we're prepping the final set of proteins that we need to make sure that this thing works. Then we jump into our mitosis, or basically the PPMAN, right? So here we go. We start lining up, condensing our DNA. So here, big thing, condense our DNA. We start copying our, so we already copied our centrosomes, and now we're gonna start pushing them apart. Our prometaphase, we're gonna start moving binding to the chromosomes, and we're gonna start organizing them, as well as like, we're setting up these, thing called astral microtubules to set up this force to kind of push each other apart. After that, my metaphase, we're gonna line up our chromosomes. Here, I think this is super important. Make sure you guys feel super comfortable with the idea that this is set up so that we make sure that we end up having the exact same copy to each cell, right? So even though when we're thinking of our regular cell in the interface, it looks like this, and there's two chromosomes, and these are homologous to each other. When that division happens, you have to make sure that each one of these ends up in the final product. And the only way to do that is that you actually keep in touch the ones that are identical, and then you pull them apart, right? So I think it's a really cool mechanism. So that's gonna be a pretty good quiz question. Anaphase, now that we know that everything's grabbed on, we're ensured that every cell is gonna be able to grab their half. You can grab on, pull them apart. Telophase, we can start building our cells again. Build the nucleus, release the DNA, and then cytokinesis, we just cut it. Everyone feeling pretty comfortable with this? Yeah, all right, so they're gonna be hidden in my slides. These yellow ones are gonna be like my study slides. If you guys can, please go back to them. Here I put, like I said, the old stuff, the old way to talk about it, the new way they talk about it, this G1S, G2M, and then what's the critical event. The big thing is I want you guys to keep a track of, for one of them is gonna be what's going on with the DNA. So make sure you guys feel comfortable kind of the questions we talked about today. The other one is this one. So here's the longer table. If we make a question that's very specific about one of the steps, this will be the big information that you guys should feel comfortable with for each one of the steps, yeah? So if my story wasn't that good today, double check my notes and then we'll back it up. All right, so for this one, there's a little bit of application on how does this actually work or what are the big effects of this. So for this one, we're talking about, there's a mutation in one of the proteins called laminin A. And what this was actually responsible for is it holds the nucleus together, right? So yeah, so it holds the nucleus together. It builds like a mesh around the outer part of the nuclear, the nucleus and the ER. So it's a big mesh holding basically just the fat bubbles in place. If you mess with that structure, during cell division, your goal is you have an organized nucleus, you have this mesh trapping it. When you let cell division start and you have to disassemble your nucleus to let this go, what they do is you basically get rid of lamellin, right? Let it fall apart. The bubbles will basically break into the smaller little vesicles and it releases the content from the inside, right? After you're done with cell division, when you have to rebuild it, again, your goal as a cell is to bring back in those lamellin, grab onto the nucleus, and then build up that structure you can hold an intact, safe environment, right? So for this, there's basically diseases that have effects in how lamellin works, right? So remember, it has to be able to hold the nucleus. It also has to be very dynamic. Like we need it to fall apart before cell division starts, and we need it to build back up right after the thing ends. So for this one, the big issue is that the lamellin itself is actually truncated, right? So we don't have that full cable that it should form, meaning that it can't actually hook onto itself to build this mesh. Because it can't do that, it doesn't make stable nuclei. So the big issue with that one there is gonna be that your nucleus is gonna be unstable. So your environment where you keep your genetic information becomes super sensitive. So that's my theory why they, because they have an unstable nucleus, the DNA is more accessible to damage. Being more accessible to damage is going to build up mutations in you that are gonna affect your ability of your cells to divide and survive. I believe with them, they have issues with aging, or it's difficult, yeah, they have really short lifespans, and it's because your cells are like super high risk of dying when it comes to trying to go undergo divisions, right? And you need to keep them going to keep our tissue alive. All right, so that's the cool thing about like one of the proteins that it does. But again, next week, what I'm really gonna chase down is gonna be this. We're gonna look at these different little places where the checkpoints help regulate the cell division. For this, we're gonna be talking about the signals, how they come in, and they help specify that a specific action has been completed from our list that we talked about today. I think we have a little bit of time, so I'll bring these up. So these are some of the cool stories of how the mechanism works. I'm gonna have you guys connect them to Friday's lecture, which is I'm gonna talk about the signal that regulates these upstream, right? So for this, I want you guys to get familiar with these two proteins. You have cohesin and condensin, right? So cohesins, they're really big, and that what they do is when you're trying to copy your DNA, right? This would be, for example, just one of these red ones. If you're trying to copy it, what it tries to do is it tries to keep that new copy to be attached to the older one, right, of these sisters. It attaches, it does it across the whole thing, and that way you make sure that you've made two identical copies and they're stuck together. At which point, there's gonna be this signaling that's gonna come in. This red thing is what we're gonna talk about next week or Friday. This kinase during the M phase, right? It's gonna end up phosphorylating the cohesin. Right? So it's gonna modify the cohesin. That modification is gonna end up having these cohesins fall off. So you need them there to help you copy the DNA. Once you're done copying it, you technically don't need them anymore. You use the kinase to kick them off. The only place they don't work is gonna be right here, where you see the centromeric region. This is where we normally attach things to help divide. In that place, we have a phosphatase. This phosphatase does the opposite, right? Doesn't let the cohesin get phosphorylated, aka it doesn't let go of the DNA. So you can keep that tight binding right at the center. At this point, you switch over to the second molecule, which is called your condensin. You have opened up your DNA initially so you can copy it, and you kept it attached to its neighbor so you knew they're identical. Once that was done, you then compress the DNA this way. And you can see that condensin, it basically compresses the chromatin into itself. At this point, you're trying to make like the most compact, shortest version of each chromosome. And you're just barely holding on to the center. So this is a really cool mechanism to make sure we copy them, and then you make these individual units. This kinetochore, I'm gonna bring it up a little bit in the end, and I'll show you how it's super functional, but let's jump to the next one. The other regulation that I really like, we're gonna go back to, it's gonna be this one, which is during the process of division, we need to break that nucleus, right? And like I said, the big thing holding it together is gonna be that lamellin. You can see here, these orange one and purple one are basically two different polypeptides, and those are the long cables I was talking about. They normally mesh up into each other, right? That mesh is what gives us that structure of like, you have a really soft plasma membrane around the nucleus, but this thing is what's giving you that mechanical force of keeping that thing intact. Here's that regulator. Here's gonna be the signaling one, CDK1 cyclin B. When this signal shows up, it's gonna phosphorylate the lamellin. At that point, it's gonna fall apart from each other, right? So phosphorylation event, they fall apart from each other, the nucleus can completely disassemble. And then the other big one that's gonna be, the other thing that's gonna be regulated or gonna be dynamic is gonna be the mitotic spindle. So for this one, actually, I need to draw an arrow here, but it's the kinases that signal, they actually work with the older microtubules. So what they'll do is they'll phosphorylate molecules that, again, the microtubule is the dynamic cable, that if you put things in the end, it'll be stable. These kinases come in and mess up the proteins that are binding these molecules at the end, right? At which point you leave this empty microtubules, they'll start falling apart. By doing that, you're gonna get rid of all the other structures, but you're gonna have a bunch of free microtubule units. So you have things available to build these new cables specifically for cell division, yeah? So big regulation up here. This is the basic stuff of how it's set up, right? Like I said, the centrosome is gonna get the minus end of the microtubule. The positive ends will basically can grow out really fast and come back. In some cases, they grow out. They can grab onto the center of the chromosomes, and we'll have one microtubule from one side, another microtubule from the other side to build this kind of connection across both sides. That ensures that we're gonna be able to put apart one copy for each cell. The other thing that we're gonna talk a little bit about is gonna be, there's these other ones called astral microtubules, here shown in red. These will be that two cables kind of come up and link up into each other like this, but there's motor proteins. The motor protein pushes this cable that way. The other copy of the motor protein pushes this cable in this direction. Right, so as the motor proteins are keep walking, they keep doing this to the microtubules. But because they behave like straws, they're actually pushing onto the centrioles. Right? So in red, you're gonna have a pushing force, and in blue, you're gonna have a pulling force. Right? So it'd be kind of like if like I was fighting with Anne for her iPad, I could be pushing against her, right, to be like, hey, get away from it. But at the same time, I could be pulling on the iPad, right? Perfect, thank you. Don't push back, because I'll fall. All right, so cool mechanisms. We'll talk about how they're regulated, and this is super fancy stuff. Like whoever engineered it, it's like you guys are on some other stuff. And this is where I'm gonna get super excited. I'm like, this feels like a quiz question, guys. Like it's that good, right? So we're gonna zoom in, right into that little pocket at the center of your chromosome. Here is where your kinetochore normally binds to the microtubules, right? And again, imagine if me and her are fighting for the iPad. For us to divide properly, she has to hold on to half of it. I gotta hold on to the other half so we can pull it apart. If only I'm holding on to the iPad and she's not holding on to it, there's basically a signal that says, hey, we haven't divided this equally yet, right? In that case, this is gonna come in. So the big player here is that microtubules are linked up to this side, so I have a copy. Because this protein complex is still there, it says that the other side still hasn't grabbed on. And the big thing that's gonna basically trap is gonna be this CBC20, right? When it's bound to it, it doesn't work, right? The moment the microtubules show up and are able to bind to it, then it can activate this anaphase promoting complex, right? That'll be the signal that, hey, this kinetochore has been grabbed on from both sides. At which point, you're gonna go to two different signaling cascades, right? If we go down the right, the big play here is gonna be separase. That's the enzyme I'm really gonna like. It gets activated, this signal goes and actually messes up with the last little bit of cohesion that we had right here. So remember, the only thing holding these things together was this last little bit of cohesion. The moment we get rid of that, this is free, right? And again, if it's me and Anne, we had that thing where it was pushing each other, but we're also pulling on it, right? So it means that she could pull her half and I could pull my half and they pull right apart. So it's a really cool pathway how we make sure we release. The other side, this is really cool in the sense of that CDK1 sigma B. So what this does is, this was the kinase. This is what was giving us the phosphorylation, like, hey, make things happen, right? Like, make this happen, and make this happen. So the moment you kick off those kinases, you're gonna have your dephosphorylation, right? You're gonna remove those phosphorylation marks. The moment you're doing that, what's gonna happen? For this one, it's we remove the phosphorylation. Now we're gonna go backwards, right? Now you're gonna have functional vanillin, and then it's gonna grab around your nucleus, boom, build a nucleus, right? So only after we've grabbed our own half of the DNA, then and only then, the mechanism set up to now we can build a nucleus. The next one. Like again, the microtubules up here, these are getting messed up by kinases, right? They're messing up any of the regular structure we have in our normal cell that are not dividing. The moment we get rid of the kinases, now we can go back to the old microtubules. We can start setting up, the old school stuff. Like you need to make up your chain below your ER to your Golgi to transport your vesicles and stuff, right? We can set that back up. And we don't have to worry about the centrioles. What else did they bring up? Condensins, nucleomelons. The Golgi kind of works like the Golgi has its own set of proteins, but they work the same way. They're there, they help link up the little cisterns of the Golgi. They get kinase function, those proteins fall off, that thing falls apart, right? Like you don't have the nice, beautiful structure. After this happens, phosphorylation marks are gone. That means your proteins can build up again. They build those nice little cisterns again. Yeah? All right, cool. So again, for this one, right below it, here I put the effects that we're picking up right now. But like I said, what we're gonna go through next week is I want you guys to, for Friday, you're gonna make the connections between how are these signals connecting into setting up each one of these pathways, right? What we're gonna see is this chart, you're gonna make it a lot fancier, and you're gonna be telling me that kind of like we talked right now, during the division phase, right? Transition from anaphase to telophase, when we're starting to pull the DNA from each other, that signal from the kinetochores, that's where the signal's gonna peak. It's gonna be low all the time, it'll peak at that point, right? When there's mechanisms that control to make sure that we've copied the DNA, they'll be low all the time, and then we'll have a peaking signal when it gets to the end of the DNA synthesis, right? So, yeah, so I don't know if it's connected like this, connected like tables, but that's the connection I want you guys to have. What are the effects of, what are the timings? What's happening to the nucleus of the DNA, the proteins? And then what's the chemical signal behind it telling us, instructing us that either things are okay, we keep going, or when something's wrong, we have to stop and wait until this thing's fixed. Yeah? Any questions? No? All right, cool. That's good. See you guys on Friday.
Y'all, I sent an email to everybody with a group link. Can y'all wanna join
I
it?
just I do get off. I feel like you're not. Yeah.
This class,